All right, ready to dive deep into signal transduction. We've got tons of research on how cells talk to each other, like cellular espionage, but with more biochemistry.
It really is amazing, these intricate communication systems. Like, think about it. Your heartbeat, your memories, even your immune system, it's all driven by these tiny molecular conversations inside your
And we're
cells.
going all the way into the mechanisms, right? We're going to be talking about receptors and G proteins and second messengers, like the whole 9 yards.
We're going to be unpacking some really cool examples, too, like how hormones, like insulin, are able to tell your cells to go ahead and take up glucose.
Yeah, and who knows, maybe we'll even figure out how some drugs actually work on like a cellular level, like beta blockers, for example. But first, I guess we should probably set the state. When we say hormones, we're basically talking about chemical messengers, right?
Yeah. Exactly, yeah. Hormones are basically like long-distance calls traveling through your bloodstream to get to their target cells all over the body. They're the key players in regulating so many different bodily functions, like growth, development, metabolism, reproduction, the whole shebang.
So imagine these hormones are like tiny little envelopes filled with instructions. How do they know which cells to deliver these instructions to?
That's where the receptors come into play. They're basically like cellular mailboxes, each one designed to recognize and bind to a specific hormone. This binding then triggers a cascade of events inside the cell that ultimately leads to a specific response.
So it's like a lock and key then. Only the right hormone can unlock that response in the target cell.
Exactly.
That's so cool.
Yeah.
But how did scientists even begin to figure out all these intricate signaling pathways?
So it was really a step-by-step process. Early on, they relied on techniques like binding assays, using radioactive hormones to track how they interacted with cells. This helped them figure out things like receptor density, which is basically how many receptors a cell has for a particular hormone.
Radioactive hormones. Wow. That's a little scary, but I guess it was all in the name of science.
It was.
So by understanding receptor density, scientists could then figure out how sensitive a cell is to a particular hormone.
Exactly. And this sensitivity can vary a lot between different cell types. Like, for example, glucagon receptors. They're super abundant on liver cells because glucagon is really important in blood sugar regulation. But you're not going to find many of those in, say, your skin cells.
Right, that makes sense. So it's like each cell has its own unique set of mailboxes designed to only receive the messages that are relevant to its job. But what about when we want to manipulate those pathways? Like when we use medications.
That's where things like agonists and antagonists come in. They're basically like molecular imposters, either mimicking or blocking the actions of natural hormones.
Oh, okay, that's interesting. So agonists first, how do they trick the cell into thinking they're the real deal?
Well, imagine an agonist as like a master of disguise. It can bind to a receptor and trigger the same response as the actual hormone would. A good example is diethylstilbestrol. It's a synthetic estrogen that was actually used to promote growth in cattle, but it later turned out to have some pretty bad side effects.
Oh, right, yeah, like the carcinogenic effects, right? It just shows you that messing with these cellular pathways can have some really unpredictable consequences.
Yeah.
So agonists are like those friends that can perfectly mimic your voice on the phone.
What about antagonists then? What's their strategy? Antagonists are like the friends that always seem to disagree with you. They bind to the receptor, but instead of activating it, they block the real hormone from getting its message through. Like, imagine them as cellular bouncers, preventing that hormone from even entering the club.
So they basically shut down the whole pathway. That's pretty powerful.
It is.
Do you have any examples of any medications that are antagonists?
Oh, yeah, definitely. Propranolol is a great example. It's a beta blocker that's used to treat various heart conditions. It actually blocks specific receptors that control things like heart rate and blood pressure.
Okay, so it's like cutting the phone lines to a specific department in the cell. I like that. But I'm kind of curious, how did scientists even figure out that receptors and adenylyl cyclase, the enzyme that makes CAMP, were actually separate things? That seems kind of counterintuitive.
Yeah, it was definitely a puzzle. They initially thought that maybe the receptor itself was adenylyl cyclase, but then they discovered that you could have multiple hormones that could activate the same adenylyl cyclase, but in different cell types. It just didn't make sense for a single enzyme to have, binding sites for all of those different hormones.
Right. It'd be like having one mailbox that could receive mail for every address in the city. So there had to be Some sort of middleman, right? Something connected the receptor to adenylate cyclase. Is that where G protein comes in?
That's exactly right. G proteins are like the couriers of the cell relaying the messages between the receptors and effector enzymes like adenylate cyclase. They're super interesting because they actually act like molecular switches, toggling between an on and off state.
Molecular switches, huh? That's pretty high tech.
Yeah.
So how do they manage to flip between those states?
It all comes down to this molecule called GTP. When a hormone binds to the receptor, it sets off this cascade that leads to a G protein binding GTP. And that's it. That's what turns the G protein on, puts it into that active on state.
So GTP is like a tiny key that flips the switch on the G protein.
Exactly.
I love that. And then what happens?
So in that on state, the G protein can then go and activate adenylate cyclase, which then starts converting ATP into CAMP.
Okay, and we know that CAMP goes on to tell the cell what to do. But how do you turn the switch off? You don't want it stuck in the on position forever.
Right. G proteins have this awesome built-in timer, you could call it. It's called GT-pace activity, and it slowly breaks down that GTP. This flips the switch back to off and stops the signal.
It's like a self-destruct button for the GTP key?
That's so cool.
Oh, so even if the hormone is still around, the signal is limited, right?
Yeah.
That must be really important so the cell doesn't overreact.
Absolutely. This regulation is it's essential for keeping things in balance inside the cell, maintaining that cellular homeostasis.
Right. But sometimes I know things can go wrong. Like when bacterial toxins mess with these carefully balanced systems.
Oh, yeah, for sure. Like cholera toxin, for example. It's a real troublemaker. It messes with a specific G protein and basically locks it into that on state. This leads to uncontrolled CAMP production, which causes, all that really bad diarrhea that's associated with cholera infection.
Oh, yikes. It just shows you how important that off switch is.
Yeah.
But can we go back to the structure of G proteins for a sec? You mentioned they have subunits, right? Can you break those down for us?
Yeah, so G proteins are what we call trimeric, which means they're made up of 3 subunits, alpha, beta, and gamma. It's this teamwork that allows them to function as those, those molecular switches we've been talking about.
Okay, so it's not just one protein, but like a team of 3 working together.
Yeah.
What does each subunit actually do?
Well, the alpha subunit is kind of the star of the show. It's the one that binds the guanine nucleotides like GTP and also interacts with other proteins to relay the signal. And to keep everything organized, it's often anchored to the cell membrane with a lipid molecule, kind of like a tether, keeping it in place.
So the alpha subunit is like the team captain, right? Making the big decisions and keeping everyone grounded. What about the beta and gamma subunits? What are they doing?
They're like the loyal sidekicks, just waiting patiently for the alpha subunit to come back from its mission. They stick together as a dimer while the alpha subunit is out there interacting with other proteins.
It's like a molecular dance. So the hormone binds, the alpha subunit grabs its GTP key, activates adenylate cyclase, the timer runs out, GTP gets broken down, and then the whole team comes back together ready for the next signal.
That's it. Yeah.
Love it. It's a remarkable system. And you said earlier that it's used in so many cellular processes, right? Not just hormone signaling.
Oh, yeah. Absolutely. Everything from vision and smell to even the control of cell growth relies on those G protein mediated pathways.
And speaking of cell growth, that reminds me, certain types of cancer are actually linked to problems with these signal transduction pathways, right?
That's right. The discovery of oncogenes, which are basically genes that can cause cancer, really revolutionized our understanding of how cell growth is regulated. They're often mutated forms of genes that are normally involved in controlling cell growth and division.
So it's like those genes have gone rogue, basically, and are now sending constant grow signals to the cell.
Exactly. And one of the first oncogenes ever discovered, it's called SRC, and it encodes a protein tyrosine kinase.
Yeah, tyrosine kinase. That sounds familiar. Didn't we talk about that before?
We did. Tyrosine kinases are enzymes that add phosphate groups to tyrosine residues on proteins. This phosphorylation acts like a switch, turning proteins on or off.
So in the case of the SRC oncogene, this tyrosine kinase is stuck in that on position, and it's just constantly signaling the cell to grow.
Yeah, that's right.
And the interesting thing is the SRC oncogene, it wasn't first discovered in human cells, right?
No, it was actually found in a virus that causes cancer in chickens.
A chicken virus.
Yeah.
So how does that relate to human cancer?
Well, that's what was so surprising. It turned out that SRC oncogene in the virus, it was actually just a mutated version of a normal cellular gene.
So the virus, hijacked a cellular gene and turned it into this cancer-causing agent. That's crazy.
Yeah, it was. And it actually led scientists to realize that a lot of oncogenes are actually derived from those normal cellular genes, which we call proto-oncogenes. And these play, really essential roles in cell growth and development.
So proto-oncogenes are like the good citizens that can be corrupted and turned into villains.
Exactly.
I like that. And then when those proto-oncogenes get like mutated or overexpressed, they can turn into oncogenes and then all hell breaks loose.
Yeah, pretty much.
Okay, this is getting pretty intense. I think we need like a little recap before we go any further.
Yeah, good idea. We covered a lot.
We did. We talked about how hormones are like long-distance messengers binding to specific receptors on the target cell to trigger those responses. And then we talked about agonists and antagonists, the molecular imposters that either mimic or block the actions of those natural hormones.
Right.
And then we went deep into the world of G proteins, the molecular switches that relay signals from the receptors to those effector enzymes, like adenylate cyclase, which then makes the second messenger CAMP.
Right.
And we even talked about how bacterial toxins can, hijack those G protein pathways and lead to diseases like cholera. And then we started talking about oncogenes, those corrupted versions of proto-oncogenes that can mess up cell growth control and contribute to cancer.
That's a lot.
It is. It's been quite a journey so far, and we're only just getting started exploring the world of signal transduction.
Yeah, just getting started. Ready to jump back into the world of cellular signaling.
Absolutely. Last time we left off talking about oncogenes and how they can lead to cancer. It's amazing how much is going on at like a molecular level. I feel like we've only just scratched the surface.
There's always more to discover, right? So let's shift gears a little bit and zoom in on how signals actually get amplified within the cell. It's not just like a one-to-one thing. It's more like a chain reaction.
I'm intrigued. So what are the key players in all of this signal amplification?
Well, we talked about adenylate cyclase and how it's involved in making the second messenger, CMP. But there's even more to the story. Adenylate cyclase itself is actually really tightly regulated by a bunch of different factors.
Oh, okay. So it's not just like sitting there passively waiting for a G protein to come along and tell it what to do.
Not at all. Remember those stimulatory and inhibitory G proteins? They're basically in a constant tug of war, and the balance between those two is what determines how much CAMP gets produced.
So G wants to crank up the CAMP production, and G wants to put on the brakes. It's like they're like negotiating over how loud the signal should be.
I like that analogy. And it gets even more interesting because there are actually multiple forms of adenylate cyclase, each with its own, quirks.
Wait, so they're like different versions of the same enzyme? What's the point of that?
Think of it like specialization. Different isoforms, different forms of adenylate cyclase can be activated or inhibited by different combos of those G proteins. So it allows for really fine-tuned control over how much CAMP gets made, depending on what the cell needs at that moment.
So it's like having a team of like specialized chefs in the cell, each one with their own like signature dish.
Exactly.
I like it. Okay, so we know that CAMP is a major player in signal transduction, but what are some of the other like important second messengers out there?
Oh, there's a whole cast of characters. Besides CAMP, there's also cyclic GMP or cGMP, and then there are calcium ions, inositol tresphosphate, and diacylglycerol. Each of these molecules has a special role in relaying and amplifying signals within the cell.
Okay, wow, that's a lot. Let's start with cGMP. Is it similar to CAMP and how it works?
Yeah, in some ways. cGMP can also activate a protein kinase. This one's called protein kinase G, or PKG. And just like pKa, which is activated by KMP, PKG can go on and phosphorylate a bunch of different target proteins, which then triggers, specific responses in the cell.
So it's like another signaling cascade, but this one's led by cGMP.
Exactly.
Okay.
Cool. But I'm curious, how does cGMP get made in the first place? Is there like an enzyme similar to adenylate cyclase that makes it?
There is. It's called guanylate cyclase, and it converts GTP to cGMP. But here's the really interesting part. It can actually be activated by a pretty unexpected molecule. Nitric oxide.
Hold on. Nitric oxide. Isn't that like a gas? I thought we were talking about enzymes and proteins.
I know, right? Nitric oxide is such a cool molecule. It can be a gas, but it can also be a signaling molecule produced by an enzyme called nitric oxide synthase. And it can just diffuse across cell membranes, acting as a local messenger.
And it's like a stealthy messenger, like slipping through walls. So what does it do once it reaches its target? Well, one of its targets is, you guessed it, guanylate cyclase. So when nitric oxide binds, it activates the enzyme, and that leads to the production of cGMP.
So it's like a gas-powered signaling pathway.
Yeah.
Very cool. And I remember you mentioned that this pathway is involved in like blood vessel dilation.
Right. Nitric oxide is a very potent vasodilator, which means it relaxes blood vessels, allowing more blood flow. And that's actually why drugs like Viagra, which boost the effects of nitric oxide, work so well in treating erectile dysfunction.
Wow, it's amazing how all these things connect back to signaling pathways. Okay, let's move on to another key second messenger, calcium ions.
Calcium ions, yeah, they're like the ultimate multitaskers in the cell. They're involved in, tons of different processes like muscle contraction, neurotransmitter release, even cell death.
Wow. So how does calcium function as a second messenger? Does it have a specific binding partner, like how CAMP binds to PKA?
One of its key partners is a protein called calmodulin. When calcium binds to calmodulin, it changes the shape of the protein, and that allows the calcium calmodulin complex to then activate a bunch of other enzymes and proteins.
Oh, so calcium and calmodulin are like a dynamic duo going around and activating other proteins to carry out their instructions.
Exactly. But this power couple needs to be kept in check, right? So the concentration of free calcium ions in the cytoplasm is usually kept very low, but it can increase rapidly when a signal comes along.
It's like a sudden burst of calcium that triggers a response. Where does all that extra calcium come from?
It can come from a couple of places. First, it can enter the cell from the outside through these special channels, or it can be released from intracellular stores, like the endoplasmic reticulum.
So it's like having both an external supply line and an internal reserve.
Okay.
Cool. But how does the cell control all this movement of calcium?
Well, the movement of calcium across the cell membrane is super tightly controlled by these channels and pumps. And the release of calcium from those intracellular stores is also regulated by specific signaling pathways. It's all about making sure things don't get out of control.
It's like a complex system of checks and balances to ensure that the calcium signaling is precise. Are any of the pathways we talked about, involved in this regulation?
Actually, yeah. One of the key players is the phosphoinositide system. We touched on it briefly when we were discussing phospholipids.
Right. It's amazing how all these seemingly different pathways are actually interconnected.
It is.
Okay, so how does the phosphoinositide system fit into this calcium signaling story?
Okay, so the phosphoinositide system involves the breakdown of this specific phospholipid called PIP2 into two second messengers. One is called diacylglycerol, or DIG, and the other is inositol 1000-4000 force phosphate, or IP3.
PIP2 gets split into DAG and IP3.
Got it. And then what do these new messengers do? Well, IP3 is the one that's really important for calcium signaling. It binds to receptors on the endoplasmic reticulum, and that triggers the release of calcium ions into the cytoplasm.
So IP3 is like the key that unlocks the calcium vault inside the ER.
Exactly. And then all that calcium can go and activate a whole bunch of other processes.
Okay, I'm following. So what happens next?
Well, this surge of calcium can activate all sorts of calcium-dependent enzymes. One of the important ones is protein kinase C, or PKC.
Another kinase. It seems like there are a ton of these enzymes that love to add phosphate groups to things.
There are a lot. And PKC is a really important one because it's actually activated by both calcium ions and DAs. And just like PKA and PKG that we talked about earlier, PKC can phosphorylate all sorts of different proteins, which influences a huge range of processes in the cell, from growth and differentiation to even regulating the immune system.
It's credible how all these second messenger systems, with their networks of kinases and target proteins, can create such a wide range of responses in the cell.
It's a beautiful system, isn't it? And what's really fascinating is that there's even more to the story than just those membrane-bound receptor systems.
Really? What else is there?
Well, not all signals actually need to go through all that trouble of binding to receptors on the cell surface and triggering all those second messenger cascades.
Wait, really? So some signals can, bypass the membrane altogether?
They can. There are certain signaling molecules, like steroid hormones, that can just slip right through the cell membrane and interact directly with receptors inside the cell.
So they're like VIPs with backstage passes. They don't have to wait in line with all the other signals. So where do these special signals go once they're inside?
So steroid hormones, because they're lipid soluble, can just diffuse right across the cell membrane and bind to receptors in the cytoplasm or even in the nucleus itself.
Wow, so they have direct access to the cell's control center. What happens then? Do they trigger some completely different type of signaling pathway?
They do. These intracellular receptors, which are often called nuclear receptors, are actually transcription factors.
Transcription factors. Those are the proteins that control gene expression, right?
That's right. So when a steroid hormone binds to its nuclear receptor, It activates the receptor, and that allows it to bind to specific DNA sequences and control which genes get turned on or off.
So steroid hormones can actually directly control gene expression. Man, that's pretty powerful.
It is. And because they're directly impacting gene expression, these steroid hormones tend to have, pretty long-lasting effects on the cell. They're not just tweaking existing proteins or pathways, they're actually changing the cell's blueprint.
It's like rewriting the cell's instruction manual.
Yeah.
Can you give me an example of it, like how this steroid hormone signaling pathway works in action? What are some of the key players in, like, the target genes?
A classic example is the glucocorticoid receptor, or GR. Glucocorticoids are a type of steroid hormone that's really important for regulating, like, metabolism, the immune system, and the stress response. So when a glucocorticoid, like cortisol, binds to the GR, it activates the receptor, which then allows it to go into the nucleus and bind to specific DNA sequences. These are called glucocorticoid response elements.
So the GR is like a courier, delivering the cortisol message straight to the cell's DNA.
That's a great way to put it. And then once it's bound to those response elements, the GR can either activate or repress the expression of nearby genes.
Okay, so glucocorticoids can either turn genes on or off, depending on the situation. What are some of the important genes that they regulate?
Oh, glucocorticoids regulate hundreds of genes involved in all sorts of processes. They can, stimulate the production of enzymes involved in glucose metabolism. They can suppress the expression of pro-inflammatory cytokines. And they can even influence the development of certain brain regions.
Wow, they really are master regulators.
Yeah.
So are there, like, other receptors for other hormones or signaling molecules that can act directly on DNA like this?
Definitely. Besides steroid hormones, there are also intracellular receptors for thyroid hormones, vitamin D, and even retinoids, which are derived from vitamin A.
So it's like a whole family of intracellular signaling pathways, each with its own, like, set of receptors and target genes.
Exactly.
It's mind-boggling to think about all the different ways that cells can receive and process signals. We've gone from membrane receptors to second messenger cascades to, like, these direct interactions with DNA.
And we're not even done yet. Remember those receptor tyrosine kinases we talked about earlier? They have their own fascinating story to tell.
Right, those are the ones that combine a receptor and an enzyme, right? I'm ready to hear more.
Well, receptor tyrosine kinases, or RTKs, they're really cool. They play a huge role in regulating cell growth, proliferation, differentiation, and even survival.
Okay, so they're involved in some pretty important processes. How do they work? What happens when a signal, like, binds to the receptor part?
When a signal, like, say, a growth factor, binds to the extracellular part of the RTK, it triggers this change in the shape of the protein. And that often leads to dimerization.
Dimerization. So that means, like, two RTKs come together. Why is that important?
Well, it's essential for activating that tyrosine kinase domain we talked about earlier. It's like they need to team up to get things going.
Okay, so it's a teamwork approach. So what happens once that tyrosine kinase domain is activated?
Then things get busy. The kinase starts phosphorylating tyrosine residues both on itself and on other proteins.
It's a phosphorylation party. So what's the purpose of all that phosphate tagging?
Those phosphate tags act like beacons, creating these binding sites for other proteins. So it's like assembling a signaling complex, bringing together all the right players into one spot.
It's like a cellular meeting point for other signaling molecules to come and interact.
Exactly. And then these signaling complexes can trigger a whole cascade of downstream events, ultimately leading to changes in, gene expression and cellular behavior. It's another example of that chain reaction with one event leading to the next.
Okay, that makes sense. Can you give me some specific examples of RTKs and like what pathways they activate? I'm curious about how all this translates into, real biological processes.
Sure. A classic example is the insulin receptor. It's essential for controlling glucose metabolism. So when insulin binds, it triggers dimerization and activation of the tyrosine kinase domain. This then leads to the phosphorylation of these proteins called IRS proteins, which go on and activate other pathways that promote glucose uptake and utilization by the cell.
So the insulin receptor is like the key that unlocks the cell's ability to use glucose. Pretty important. What about other RTKs?
Well, RTKs can activate a whole variety of different signaling pathways, including the PI3K ACT pathway, the RAS-MAPK pathway, and the PIL-SALT pathway.
Okay, that's a lot of pathways. What are those pathways all about?
Okay, so the PI3K ACT pathway, it's involved in things like cell growth, survival, and metabolism. The RAS-MAPK pathway, that one's involved in cell proliferation and differentiation. And the PLC pathway, that's involved in calcium signaling and the activation of PKC, which we talked about earlier.
Wow, it really is like a tangled web of interconnected pathways.
It is, and these pathways are often dysregulated in cancer, which highlights how important it is to understand how RTKs work and what happens when things go wrong.
This is all so fascinating. It's incredible how these tiny molecular switches with their intricate networks of phosphorylation and signaling complexes can have such a huge impact on our health. But I think we need to zoom out a bit again and talk about how all of these different signaling pathways are coordinated at a whole body level.
You're right. It's important to see the big picture. We've been focusing on individual pathways, but they don't operate in isolation, right? They're all part of a larger interconnected system.
Right, of course. So where do we start? Let's go back to hormones. I know that in vertebrates, there's a sort of hierarchy of hormonal control.
That's a great place to start. Understanding the hierarchy of hormonal control helps us really appreciate how different organs and systems communicate to maintain homeostasis.
Right, homeostasis,
Mm.
keeping everything running smoothly. So where does this hormonal hierarchy begin?
It all starts in the brain with the hypothalamus. It's a small region, but it's super powerful. It's often called the master gland. The hypothalamus acts like a central command center, receiving signals from both the brain and the body, and then it releases hormones that control the activity of the pituitary gland.
So the hypothalamus is like the CEO of the endocrine system.
Exactly. It sends out instructions in the form of these hormones called releasing factors, which travel to the pituitary gland. These releasing factors can either stimulate or inhibit the release of pituitary hormones.
Okay, so the hypothalamus is the boss, telling the pituitary gland what to do. What does the pituitary gland do then?
The pituitary gland is also sometimes called the master gland, and it releases a whole variety of hormones that regulate the activity of other endocrine glands, like the thyroid gland, the adrenal glands, and the gonads.
So it's like the middle manager, relaying instructions from the CEO down the chain of command.
Exactly. And those pituitary hormones have a wide range of effects on the body, regulating everything from, metabolism and growth to reproduction and stress response.
So it's like this chain of command where each level regulates the one below it.
That's a good way to think about it. And that structure makes sure that hormone release is really tightly controlled, preventing any overproduction or underproduction of those powerful signaling molecules.
This is fascinating. But what about those feedback loops we've mentioned a few times? Those are important for keeping things balanced, right? How do they fit into this picture of hormonal control?
Feedback loops are absolutely essential for keeping those hormone levels within a very narrow range, preventing any imbalances that could really mess things up.
So they're like a thermostat, making sure the hormonal environment is just right.
That's a perfect analogy. There are two main types of feedback loops: negative feedback and positive feedback.
Negative feedback is the one we hear about most often, right? It's like hitting the brakes when things start to get out of control.
Exactly. With negative feedback, the release of a hormone eventually leads to a decrease in its own production.
Okay, can you give me a specific example of how that works?
Sure. Think about cortisol. It's a stress hormone produced by the adrenal glands. So the hypothalamus releases corticotropin-releasing hormone, or CRH, which then stimulates the pituitary gland to release adrenocorticotropic hormone, or ACTH. And then ACTH travels to the adrenal glands and tells them to make cortisol.
Okay, so it's like a three-step process. Hypothalamus releases CRH, pituitary releases ACTH, and then adrenal glands release cortisol.
Exactly. But here's where that negative feedback loop comes in. High levels of cortisol in the blood actually inhibit the release of CRH and ACTH from the hypothalamus and pituitary.
So cortisol is basically telling the hypothalamus and pituitary, hey, I'm already on it. No need to send out any more signals.
That's a great way to put it. And that feedback system is what makes sure that cortisol levels don't go through the roof.
It's like a self-regulating system.
Very cool. Okay, so that's negative feedback. What about positive feedback?
Positive feedback is a little less common, but it's still important. It's more like a snowball effect, where the release of a hormone leads to an increase in its own production.
So it actually amplifies the signal instead of dampening it. When is that useful? Positive feedback is really important for processes that need to happen quickly and powerfully, like childbirth and blood clotting.
Oh, okay, so it's like slamming on the gas instead of the brakes.
Yeah.
Can you walk me through how positive feedback works during childbirth?
Sure. During labor, as the baby's head starts pushing against the cervix, it triggers the release of oxytocin from the pituitary gland. Oxytocin then causes the uterus to contract, which pushes the baby's head even harder against the cervix, and that stimulates even more oxytocin release.
It's like a cycle that just keeps reinforcing itself, leading to stronger and stronger contractions until the baby is finally born.
Exactly. That positive feedback loop is essential for a successful delivery.
Wow. It's incredible how these feedback loops, both positive and negative, are so important for regulating hormone levels and keeping everything in check. But let's change gears a little bit and talk about how peptide hormones, which are another major class of messengers, are actually made.
Peptide hormone synthesis is a really fascinating process. It's a multi-step thing and involves gene transcription, translation, post-translational modifications, and then finally secretion.
Okay, that's a lot of steps. Let's break it down. So what's the role of gene transcription and translation in making those peptide hormones?
Well, like all proteins, peptide hormones are encoded by genes. So the first step is transcription. That's when the DNA sequence of the gene is copied into an mRNA molecule.
So it's like making a blueprint for the peptide hormone.
Exactly. And then that mRNA molecule goes out of the nucleus into the cytoplasm, where it gets translated into a protein.
Okay, so the mRNA blueprint is used to assemble the peptide hormone.
That's right. But here's where it gets interesting. A lot of peptide hormones are actually synthesized as inactive precursors. We call them pre-prohormones.
Pre-prohormones. Why are they made in an inactive form?
These pre-prohormones have a special signal sequence that tells them to go to the endoplasmic reticulum, where they undergo all these modifications. It's like a finishing school for those peptide hormones.
So it's like they're sent off to get, polished and refined.
Exactly. In the ER, that signal sequence is chopped off, and the pre-prohormone gets converted to a prohormone, which is getting closer to its final active form.
Okay, what happens to the prohormone next?
From the ER, the prohormone is transported to the Golgi apparatus. That's another cellular compartment involved in, protein processing and sorting.
So it's like a packaging and distribution center for newly made proteins.
That's a great way to put it. And in the Golgi, the prohormone can undergo further modifications, including something called proteolytic cleavage. And this cleavage can actually create multiple active hormones from just a single prohormone.
Wait, you're telling me that one prohormone can give rise to several different hormones? That's amazing.
It is. It's a super efficient way for the body to make a bunch of different signaling molecules from a limited number of genes.
It's like a molecular assembly line, making all these different products from the same starting material. Okay, that's cool. But once these peptide hormones are all processed and packaged, how do they actually get out of the cell into their target tissues?
They're usually stored in these things called secretory vesicles inside the cell. And when the cell gets the right signal, these vesicles fuse with the cell membrane and release those hormones into the bloodstream.
It's like a cellular delivery service, sending those hormones exactly where they need to go.
Exactly. And just like every other step, this secretion process is really carefully regulated to make sure that the right amount of hormone gets released at the right time.
It's mind-blowing how much coordination and precision goes into making, processing, and delivering those peptide hormones. Are there any examples of those multi-hormone precursors that produce hormones that have totally different functions?
There are. A really fascinating example is pro-opiomelanocortin, or POMC. It's a big precursor polypeptide that can actually give rise to several different hormones, including adrenocorticotropic hormone, which is ACTH, melanocyte-stimulating hormone, or MSH, and beta-endorphin.
Wow, that's quite a mix. ACTH is involved in the stress response, MSH influences skin pigmentation, and beta-endorphin is a natural painkiller. That's crazy that they all come from the same precursor molecule.
I know, right? It's really cool. And what's even more interesting is that POMC can actually be cleaved at different spots depending on what tissue it's in. So you end up with different sets of hormones being produced in different tissues.
So it's like a molecular toolkit. Different tissues can use different tools from that toolkit to make the hormones they need.
Exactly. This tissue-specific processing of POMC just shows how adaptable and versatile peptide hormone synthesis can be. But we've been talking about hormones for a while now. Remember that link between signal transduction and cancer that we talked about earlier? Maybe it's time to circle back to that.
Oh yeah, you're right. That's a really important part of the story. When those signaling pathways get disrupted, things can go really wrong. Let's dive back into the world of oncogenes and see how they mess up those cellular conversations.
Sounds good to me.
So let's pick up where we left off talking about oncogenes and how they can mess up cellular communication and lead to uncontrolled cell growth.
Right. Remember we were talking about how oncogenes are basically corrupted versions of those proto-oncogenes, and how proto-oncogenes usually have a role in regulating cell growth and division.
Right, and proto-oncogenes often encode proteins involved in signal transduction pathways, right? Like those growth factor receptors and protein kinases and even transcription factors.
Exactly. So
Oh,
when those proto-oncogenes get mutated or overexpressed, that's when they turn into oncogenes and can really mess up those carefully regulated signaling pathways.
and I remember we talked about how one of the earliest oncogenes discovered was SRC, and it encodes a protein tyrosine kinase.
Right.
And tyrosine kinases add phosphate groups to proteins, acting like switches that can turn processes on or off.
That's it. And with this RC oncogene, that tyrosine kinase is stuck in the on position, constantly telling the cell to grow.
It's like having the gas pedal stuck to the floor.
Yeah.
But wasn't SRC first discovered in a pretty unexpected place?
It was. It was actually found in a virus that causes cancer in chickens.
A chicken virus?
Yep.
Wow. So how does a chicken virus relate to human cancer?
Well, that's what made the discovery so groundbreaking. It turned out that the SRC oncogene in that virus was actually just a mutated version of a normal cellular gene.
So it's like the virus stole a cellular gene and turned it into this cancer-causing agent. That's incredible.
It was a huge discovery. And it led scientists to realize that lots of oncotrusts are actually derived from those normal cellular genes, those proto-oncogenes we were talking about.
Right, like good citizens being turned into villains.
Exactly. And it's important to remember that these proto-oncogenes are actually crucial for normal cell growth and development. It's only when they are mutated or overexpressed that they become dangerous.
So it's all about balance.
It is.
But are there other ways that viruses can contribute to cancer besides just hijacking proto-oncogenes?
Yeah, absolutely. Some viruses can actually insert their own genetic material right into the host cell's DNA.
And that can cause problems.
It can. That viral DNA can disrupt the function of nearby genes, and that includes tumor suppressor genes.
And tumor suppressor genes are the good guys, right? They help prevent cancer.
Exactly. They encode proteins that act like brakes on cell growth, making sure things don't grow out of control.
So if a virus messes with a tumor suppressor gene, it's like cutting the brakes on cell growth.
That's a really good analogy. And that loss of control can make someone much more likely to develop cancer.
Wow, it sounds like viruses have a whole arsenal of tricks. But besides viruses, what other things contribute to cancer?
Well, cancer is complicated, and it's usually a combination of genetic and environmental factors that cause it.
So it's not just about our genes, but also about the world around us.
Exactly. Exposure to certain chemicals, radiation, and even lifestyle factors like smoking and diet, and even lack of exercise can play a role.
Right. It's a good reminder that our health is this complex interplay of nature and nurture. But speaking of health, I know that understanding signal transduction is crucial for developing new therapies, especially for diseases like cancer.
Definitely. One promising area of research focuses on developing drugs that target specific signaling pathways that are messed up in cancer cells.
So trying to disrupt those corrupted lines of communication that are driving the uncontrolled growth.
Exactly. For example, there are some cancer drugs that target particular tyrosine kinases that are overactive in certain types of cancer. And by blocking those, we can slow down or even stop tumor growth.
It's like using our knowledge of these pathways to create more precise and more effective treatments. That's amazing.
But before we wrap up our deep dive into signal transduction, I want to touch on neurotransmission again.
Yeah, that's a good idea. We've talked about it briefly before, but neurotransmission is a great example of how those signal transduction principles are used in the nervous system, letting neurons communicate quickly.
Okay, remind me, how does neurotransmission work?
So it starts with the release of those special chemical messengers, neurotransmitters from a neuron, which we call the presynaptic neuron. And those bind to receptors on another neuron called the postsynaptic neuron.
So it's like a cellular conversation with the neurotransmitters acting as the words.
Exactly. And these conversations are happening all the time throughout your nervous system. They're how you're able to think and feel and move and experience the world around you. That's amazing. And I remember you mentioned there are two types of synaptic transmission, fast and slow.
Right. Fast synaptic transmission is all about speed. It uses ion channels that open and close quickly, changing the electrical potential of that postsynaptic neuron. This happens super fast, like in milliseconds, so it allows for those really quick responses, like reflexes.
So it's like sending a text, instant communication.
Exactly.
What about slow synaptic transmission then?
Slow synaptic transmission is more like sending an email. It involves activating those intracellular signaling pathways, the ones we've been talking about, and often uses second messengers. It takes a bit longer, maybe hundreds of milliseconds or even minutes, but the effects last longer.
Okay, so fast transmission is for those quick reactions, and slow transmission is for more complex, longer-lasting responses. What are some of the key neurotransmitters involved in that slower type of signaling?
Well, lots of neurotransmitters can be involved in slow transmission, but some of the most well-studied are dopamine, serotonin, and norepinephrine. They're really important for regulating mood, motivation, attention, and even things like learning and memory.
It's incredible that these tiny molecules can have such a huge impact on our thoughts and feelings and behavior. But how do drugs like antidepressants and antipsychotics work?
Lots of psychopharmacological agents actually work by targeting specific neurotransmitter systems. So, for example, some antidepressants, like Prozac, they work as selective serotonin reuptake inhibitors. Those are called SSRIs.
SSRIs. I've heard of those. How do they work?
Okay, so normally after a neurotransmitter is released into the synapse, it can be taken back up into that presynaptic neuron, like it's being recycled, and that helps regulate how much of the neurotransmitter is in the synapse. SSRIs block that reuptake process, specifically for serotonin.
So it's like preventing the cleanup crew from removing serotonin from the synapse, leading to higher levels of serotonin signaling.
Exactly, and that increase in serotonin is thought to be why SSRIs can improve mood.
That makes sense. But what about other drugs? Are there ones that target neurotransmitter systems in different ways?
There are. Some drugs target the enzymes that actually break down neurotransmitters. Like MAOIs, a type of antidepressant, they block the enzyme monoamine oxidase, which breaks down neurotransmitters like dopamine, norepinephrine, and serotonin.
So MAOIs protect those neurotransmitters from being destroyed.
Yep. And that increases the levels of those neurotransmitters in the brain.
It's amazing how these drugs can fine-tune neurotransmitter levels to help treat mental health conditions. But before we wrap up, I want to mention one last group of signaling molecules: peptide neurotransmitters and neurohormones.
Right. These are a really unique group. They can act as both neurotransmitters and hormones, and they often have lots of different roles in the nervous system.
Like what?
Well, they can influence things like pain perception and stress response, as well as appetite and social behavior.
So many different roles. Can you give me some examples?
Sure. One really interesting group is the endorphins. There are natural painkillers.
Endorphins, like the runner's high.
That's it. And they were actually discovered when scientists were studying opiate drugs, like morphine.
So endorphins are like the body's own morphine.
In a way, yes. They bind to the same receptors in the brain as those opiate drugs do, which is what gives you that pain relief. That's why your body releases endorphins when you're stressed or in pain to help you cope.
It's like we have our own built-in pain relief system.
Yeah.
What about other peptide neurotransmitters and neurohormones?
Well, they're involved in all kinds of things, from sleep and appetite to the stress response and social behavior.
Wow, it's amazing how these tiny peptides can have such powerful and diverse effects.
It is. And scientists are still researching peptide neurotransmitters and neurohormones, making new discoveries all the time. They're even exploring their potential as therapies for things like chronic pain, anxiety, and even addiction.
It's been an amazing journey through the world of signal transduction. We've covered so much, from how cells receive signals to how they process them and respond, talking about hormones and neurotransmitters and growth factors.
And we've talked about all those amazing molecular machines that make these pathways work, like G proteins and second messengers and protein kinases and transcription factors.
And we've seen how all of these pathways are connected. This amazing network that's controlling pretty much every aspect of our biology.
It's truly incredible.
We even touched on how those pathways can be disrupted, leading to diseases like cancer. But all those challenges bring opportunities, and now that we're understanding more about signal transduction, it's leading to all these new and innovative therapies.
Definitely.
It's mind-blowing how all those tiny molecular conversations happening in our cells are shaping our health, our behavior, everything about who we are.
I know. It's pretty amazing.
So next time you feel your heart racing or your stomach growling, or you just have a random thought, remember that invisible world of signal transduction working away inside you.
That's right.
Thanks for joining us on this deep dive into signal transduction. It's a truly fascinating field, and I hope you learned something new.
I did too. It's always fun to explore these topics.
And as always, keep asking questions and stay curious because there's so much more to discover.